Okay, so let's get started then. All right, so cell history and theory. No, he's like, ooh,
I like that.
what do you have there? This is a microscope. Wow, that's very interesting. I found this. That's what he said. I found this. I have no idea what it is.
He's like looking off at the stars.
Okay, so we saw with our section on macromolecules, the carbohydrates, the lipids, proteins, and nucleic acids. We mentioned with each one of those larger molecules that they are built of smaller building blocks, monomers, that make up the polymers. And so life is much the same way, where we have a monomer, the cell, that all life is based upon. Like the cell is the smallest unit that has all the criteria for being a living thing. All right, so it's that basic unit for all forms of life. And so to be alive, it must be made of cells. If the cell is the monomer, then anything we see that's living must contain cells. Atoms and molecules are not alive because they do not show all the different properties of a living organism. They do not require nutrition or nourishment. They do not, they don't reproduce. So they don't really fall into that idea of being alive, right? So these guys, though, we didn't really recognize them as the building blocks of life until after the invention of the microscope. And so those gentlemen were all clustered around a very crude, rudimentary made microscope. Thank you, sir. And so we're probably looking at that picture was probably taken around 1655. Yeah, that picture in color, by the way. Yeah, I know. Really great. No, the illustration is probably showcasing some gentleman held around a microscope around 1655. All right, so the cork cells then were first seen by, published anyway, by Robert Hooke in 1655. So in 1655, the microscope had just kind of been thought up. Rather than looking at using a long tube with some ground lenses to look at the heavens, we try to reorient and reconfigure those lenses to look at things that are very, very small in front of us or near us. And so using a crude microscope, he looks at this section of dead wood, that medium between the actual outer bark of the tree and the inner living part of the tree, those cork cells. And these empty boxes or cells, he coined the term cell because it reminded him of the cells in the local monastery. This monastery has a bunch of different small holding rooms or cells, and so he thought that this was very much like that in structure and appearance. So this observation that things are made of these small cells gave rise to cell theory. Right? So the cell theory has three main parts. All right, obviously the first one is simple. All living organisms are made of cells. Right? If the cell is a natural building block of life, then everything that's living must have them. The second point then, that cells are the basic units of structure and function in living things. So even in large organisms like ourselves, everything that we have, everything that makes us up, is contained within cells, and those cells have different functions and features and structures based upon what they are supposed to do. And then the third point is one that probably caused the most controversy, especially in the late 1600s, and still causes a lot of controversy today, a lot of discussion. It's kind of opening a can of worms a little bit when you mention this about cell theory, but that new cells come from existing cells. Why would that be a problem? Why would that be a controversial statement, or why would that be a statement that you're kind of asking for trouble with? Yeah.
Because where did the first one come
from? Absolutely, right to the point. Where did the first one come from? Right. If all cells come from other cells, then where did the first one come from? That originating first cell. We've seen lab experimentation where they take a primordial soup of different chemicals that would have existed on Earth early in its formation, and given it some extra energy like electric sparks or shock or something. And we've seen the spontaneous, not spontaneous, but the formation of simple amino acids. And we know that amino acids, when put together into a chain, make proteins. So, and that's really, at the heart of it, life moves from genetic information to protein. We have seen, or we know, that we can take phospholipids, we can put them into an aqueous environment, and they will line up with each other, making like a very rudimentary membrane. Or we can even put them together where they will, on their own, form into these spheres, locking water outside and preventing water from getting in. So this is kind of cool stuff to think about and consider looking at all the information. All right, so there's all kinds of different cells, right? We look around us, we have all kinds of different living organisms, and so therefore there are different types of cells, and we know that cell structure is related to its function. What it looks like really helps us understand what it might be used for. All right, so we have a very similar image like the plant cells we had seen at the beginning, and we can kind of think about like the fact that these strong cell walls and their boxy shape really help form, one, the shape of the overall organism, but two, that it's very strong and rigid and structurally sound as far as its function. So nerve shape, tree-like branching and its function send those electrochemical messages from one to the next. Right? It's almost like telephone wires almost. Its structure, again, kind of dictates its function. The red blood cells, their shape, they're like a sat-on beanbag. They have this biconcave shape where they're kind of squished in the middle, and if you were to turn them on their side and cut them in half, they look like a weird looking dumbbell, like that. All right, and the reason they look like that is, well, their function. Their function is to transport oxygen to all parts of our body, and they can do that. They can squeak through little capillaries by their shape. Right, so they don't clog up and get jammed up trying to move through those small little capillaries. And then finally, like skin cells. We know that the dermal layer, the living layer of skin cells, are kind of more of a boxy square shape. But as they move their way out and die and become the epidermis layer of our skin, they kind of get squished and stretch out, and they form a really good barrier between the outside environment and the inside of our bodies. So 3 good examples where the form really does give away its function. So what are the main categories of cells? There's only 2 main categories in the entire world. So this makes our life as a cell biologist really easy. Right? Either you're a prokaryote or you're a eukaryote. So let's take a look at each. All right, so prokaryotic cells, if you want to think right away bacteria. Bacteria are prokaryotes. All right, you can easily sketch in your mind or even sketch on a page a very simple rod-shaped bacteria, and we'll throw a flagella on it too, just because. Why not? Because it needs to move around. It might have some cilia or maybe some micropilli hanging out around it as kind of receptors and stuff that it can kind of feel its way around and know what it's next to. But these do not contain a nucleus. Right, their genetic information is actually just kind of sprawled out in the cytoplasm, and it's like a circular loop. So it's like a big, long, ginormous rubber band that you've kind of stuffed into a box. It's all kind of coiled on itself and wrapped up on itself. But if you were to take it out and make it nice and neat, and lay it out nice and flat, and make a big loop. Right, that's what we have here inside these bacteria. It's prokaryotes. Right, there's no membrane-bound organelles. So you're not going to find, like mitochondria or chlorophyll or chloroplasts. Sorry, you're not going to find chloroplasts because they just do not exist at the prokaryotic level. Right, and they're usually smaller than eukaryotic cells. Their size ranges between 1 to 10 micrometers. All right, so the oldest cells known then are prokaryotic cells that we find in like kind of impressions or fossil remains, looking at them at the microscopic level. And they've been dated to around 3.5 billion years old. We talked about Luca before, the last universal common ancestor. And we talked about it in terms of like the hominids, like the Homo sapiens, like well, that branched out from the different hominid. If we were to look at like all life on Earth and everything's last universal common ancestor, we should, in theory, be able to trace it all the way back to a prokaryotic cell. That's the first living thing on this planet that had all the qualifications of being living was a simple, single-celled prokaryotic cell. Simple bacteria or something. All right, so how do we get from a prokaryotic cell to a eukaryotic cell? All right, well, we're going to suggest this idea of endosymbiosis. Right? We go from prokaryote to eukaryotic cells through this idea. Right? So, in a nutshell, in a very simplified way, we have two independent bacteria. Right? One a little larger than the other one, and the larger one, it's like Agario, right? It's like the bigger one, like engulfs the smaller one. Boop, it becomes part of it. And so now we have a bacterium that's living within the larger one. Now, rather than digest it and break its membrane down and destroy it, what we find out is maybe this bacterium is actually resistant to that, but it's actually able to like maybe produce energy, and it produces extra energy that it gives to its host, right? That it gives to the bacterium that's living within. So we have this mutualistic setup where both benefit from hanging out with one another, right? So they're both bacteria. So within the larger one, the smaller bacterium is replicating, it's duplicating itself. It's going through binary fission, making copies of its simple circular DNA, and then pinching off, and bloop, there's another one. Bloop, there's another one. So there's a bunch of these guys start to hang out inside this larger bacteria. The larger bacteria doesn't quite duplicate as often as the smaller one, but when it does, and it pinches in half and bloops one of itself, right? It includes some of this bacteria that it had formed this endosymbiotic relationship with. And that continues to be passed on from generation to generation. Right? This is where they came from and how they came to be part of our cells, the eukaryotic cells. All right, so if we have one more slide here, this image is a little bit more clear-cut. There are three major steps that got us to what we consider to be like our modern eukaryotic cells. Right? The first thing that happened was we had the cell membrane of this proto-eukaryote. Not quite a eukaryotic cell yet, but on its way to be. There was so much extra like cell membrane that it kind of started to fold up a little bit on itself, and then it became kind of part of the interior of the cell. And what we found to be advantageous is that if we actually surround our genetic information with a protective layer, its own little membrane. And so this is going to give rise to the formation of the nucleus, and then some of the extra foldings, this like tube-like cavernous structure that's kind of left over, became the endoplasmic reticulum, or the ER. All right, so your nucleus and your endoplasmic reticulum formed first. So now we have a nucleated cell. All right, the second event that happened was the first endosymbiotic event, where we had a nearby bacterium, right, actually learned or knew how to respirate and go through cellular respiration. So it was an aerobic bacteria, one that could use oxygen to make simple sugars and stuff, or use oxygen, to break down simple sugars. We incorporate that into our cell, into this proto-eukaryote, and they eventually become the modern-day mitochondria. Right? So, because now we have this simple bacteria that is able to take oxygen and simple sugars and make a crap ton of energy. So much energy that it doesn't need it all. That it can give it to the larger eukaryotic cell that's within. Right? So this becomes that membrane-bound organelle, the mitochondria. The third event and the second endosymbiotic event meaning that we're bringing a bacteria into the cell and it becomes part of the cell later on as an organelle, would be the photosynthetic bacteria. All right, so another bacteria, so we already have eukaryotic cells with mitochondria. Then we have some that absorb in or bring into themselves ones that can actually photosynthesize. Right? Those become our chloroplasts, or the chloroplasts. So when discussing when this kind of happened or the order of events, we know that the mitochondria had to be brought in first because plants have mitochondria and chloroplasts. We just have mitochondria. You know, what we see from prokaryotic cells to eukaryotic cells, this theory really has the most evidence and facts to kind of support it. Because not only do we see like mitochondria in eukaryotic cells, and we see these chloroplasts in eukaryotic plant cells, the chloroplasts and the mitochondria have their own DNA. Like mitochondrial DNA is the DNA of the mitochondria, and it has its own ribosomes, and it expresses its own proteins, and it can make its own energy. It doesn't need to be in our cells to exist, but it's there. Chloroplasts, same way. Chloroplasts have their own DNA. Okay, so eukaryotic cells have the nucleus and the membrane-bound organelles. Compared to prokaryotic cells, obviously the eukaryotes are much more complex. We consider them to be evolved from prokaryotes, and these eukaryotes include protists, animals, plants, and fungi. Viruses, but these are not cells, okay? Viruses are not living things. The reason is they do not reproduce on their own. They require a host to reproduce or make more of their genetic lineage, I suppose. Right? They will contain DNA or RNA depending on what's inside this capsid here. It's a good way to kind of really quickly check yourself over to make sure that you can pick out what parts prokaryotes have, eukaryotes have, and viruses, well, usually don't have a whole lot. Some common cell structures then, all right, are things that are common from one cell to the next. All cells, both prokaryotic and eukaryotic in nature, carry out three tasks, right? Really, these are kind of like the living tasks almost that all cells have to do, or organisms have to do. One, they have to find food and nutrition and make that into energy, right? So they need to eat. Right? They also, if they're going to eat and process stuff, they got to poop. Okay, so they have to get rid of waste. Otherwise, it'll be like one big garbage bag. It's bigger and bigger and bigger, and it's no good. Right? It would not end up being a good day. Right? And then finally, making more of themselves. Right? So reproduction. And that's really what living things are on this planet to do. Eat, poop, and make more of themselves. You add our higher level reasoning to that, and then you add societal structures, and now like you need to get an education and get a job and make money and exchange currency for goods and services, fall in love, have a family, all that stuff. Not necessarily in that order. Some orders are easier than others to do, so make smart decisions. All right, so four parts of cells that all cells have in common. All right, they're going to have a plasma membrane or cell membrane. Okay, that makes them a cell, easy. Within that membrane, they're going to contain some liquid goodies, cytoplasm. They're also going to need ribosomes to express that DNA, and then from our DNA to RNA to a protein. All right, so we need to be able to do that. And finally, that genetic information. We need the instructions. Right? So they need to have DNA as well. All cells have these four parts in common. Una meaning one. All right, so organisms that are made of only one cell have to do all of those features. You know, eat, feed, or eat. We can combine those. We can call it eating. Right? They have to be able to get rid of that waste, and they have to reproduce. Okay. They have to do all that all in one membrane. This includes all prokaryotic cells. Like all prokaryotes are single-celled organisms. Think bacteria. Right? It's the easiest go-to. This includes also some eukaryotic cells, like some fungi, not all, but some fungi are unicellular, as well as some protists. The example for the protists would be like blue-green algae. For the multicellular organisms, it's more complicated, but it's made of more than one cell, and therefore the cells can specialize, right? Certain cells will do certain tasks based on what their specialization is and contribute to the survival of the entire organism. So if you have this multicellulated organism with these specialized cells, that will lead to a hierarchy of organization, right? And we've mentioned this before, but it bears repeating. If we have a bunch of cells that team up to serve a common purpose, then we form some tissues with those cells that work together with a common function. The example we can go to would be smooth muscle tissue. A bunch of smooth muscle cells that work together, and the smooth muscle is more for like very strong but sustained contraction and relaxation of that tissue. If we take a group of tissues and fashion them together to work for a common purpose, then we form an organ, right? And that organ, then we could use the example of the stomach. A group of organs working together will then make that a system. So this should not be new to you. This should simply just reinforce the thought that if you have cells that specialize for a certain job, then a group of them will form a tissue, a group of tissues will form an organ, a group of organs will form the system.